Let's now look at how specific groups of animals deal with osmoregulation, and we'll start with the invertebrates. So marine invertebrates in general are osmoconformers. So most all marine invertebrates have an osmotic concentration in their bodies that matches the saltwater around them. Most freshwater invertebrates are osmoregulators, so they can't be pure fresh water. They need to maintain the extra ions and solutes in their bodies, so they have to regulate that. And the main stress of being a freshwater invertebrate as it relates to osmoregulation is gaining water. Since the environment that they live in is hypoosmotic, they are constantly taking on water and have to deal with that extra water. And then terrestrial invertebrates, most of them are osmoregulators, and their main stress is losing water, water evaporating from their bodies into the atmosphere around them. All right, so one means of osmoregulation in vertebrates is using contractile vacuoles. So this is individual cells sort of trapping extra water and then expelling that water through exocytosis. And there are some freshwater sponges that use this, constantly taking on fresh water, and then they expel that water through these vacuoles. So here are some cells that are doing that. Those big circles in the middle of the cells represent those vacuoles full of water. And this is an image of one of these freshwater sponges that takes advantage of this mechanism. Some invertebrates use protonephridia, so this is a sort of rudimentary kidney-like structure in the animal. And this includes flame cells. These are specialized cells, just named flame cells because they kind of look like they have a flame coming out of them to remove the water. And these flame cells are found in annelids, the segmented worms, platyhelminths, the flatworms, and some molluscan larvae. So here we have a free-living flatworm, and they've got these flame cells you can see sort of blown up in the drawing on the right. And these flame cells will help expel water from their bodies. Another mechanism is through metanephridia. So metanephridia are a little more complicated than protonephridia. These are tubules that will draw water out of their bodies. And there are some annelids that have metanephridia, and most molluscan adults take advantage of this. So this is from an annelid, a segmented worm. You can see the repeated segments, and then those blue tubules are going to represent these metanephridia, and they're able to extract water from their bodies and expel it through these tubes that end at those nephritiopores. And some other animals will take advantage of antennal glands. So these are glands near antennae, and they will allow hemolymph, so that's the fluid that circulates in their open circulatory system, to enter into these canals, these nephridial canals. And then the ions will get reabsorbed, so the water with the ions gets expelled, the ions then get pumped back into the body, leaving the water behind. And these are found in freshwater crustaceans. So here's a picture of a crayfish, and you can see in green there the antennal glands, allowing body fluid, hemolymph, to pass into this tubule, and then the ions get pumped back into the body, leaving the water behind. There are also Malpighian tubules. So this is another mechanism used by invertebrates. Ions get secreted into tubules. This is a way to get rid of excess ions. This is found in insects, and some arachnids take advantage of this as well. So this is along the digestive system. There are these little tubes sort of branching off of the digestive system. You can see it sort of in the middle of the drawing here, the Malpighian tubules. And these tubules will then excrete, pump ions into those tubule lumens, can allow water to pass in there too, and then they can reabsorb whichever of those they need to. So they can kind of play with the ratio of these to get rid of excess ions if needed, or water. Some animals have coxal glands, and this is where waste gets secreted into sacs. And this is found in some arachnids. So kind of similar to Malpighian tubules functionally in a way. So we could see in this drawing of an arachnid, there's these coxal glands that'll be able to secrete waste.